in the intersection between epigenetics, aging, and exercise. We know that aging is accompanied by decline in functional capacity across the lifespan. And in fact, across every organ system, age stands out as the single biggest driver of disease. And as you can see here, all individuals will experience changes with chronological aging, but some individuals will have different biological age. And what it means is that their functionality will decrease earlier in life, and they will lose their health span. And health span, and this is what we need to focus on, is the number of years that we live in good health. So instead of just elongating life of people, which is accompanied by health span, we need to make sure that they spend in the 10 or 15 last year of their lives in good health. So this is the sort of the entire mission of the aging research. And as you can see here, some people will, have their biological aging plateau for many, many years in a good health, when some adults are declining very quickly. And one of the hallmarks of aging is epigenetics. So what is epigenetics? Epigenetics are simply modifications in our genome that affect the expression of the gene. So these are modifications that, as a consequence, the gene expression is changing. And epigenetic changes stand out as one of the hallmarks, the big hallmarks of aging. Telome, as I said, is dynamic, and CpG sites can be added and removed by DNA methylation enzymes. And many CpG sites has been shown to change with age. So as you can see here, this is birth and age, and you can see this is a specific CpG site across the genome, and it can be unmethylated when we're young and then get methylated across the genome when we're getting old. The interesting thing is that we are looking at that, or we're measuring CpG sites on a gradient. Although it's a binary thing, either methylated or not, we are measuring that on a gradient of 0 to 100% across the genome, across the cell population. So because we are very interested in exercise, we wanted to answer a few questions. First of all, do fitter individuals, people with higher fitness at baseline or lower fitness with baseline, display younger methylation and transcriptome profile than less fit individual? Now, we also looked at transcriptome. As I said before, the DNA methylation is going side by side with gene expression, and changes in DNA methylation are often very highly correlated with changes in the transcriptome. So we looked at both at baseline. Then we looked at exercise. So we wanted to ask, does exercise training mitigate or reverse omic aging in muscle aging, in human muscles, sorry. So data sets with exercise training. And then we looked at, I guess, the other end of the spectrum, which is immobilization data that we had to look, and which we think should be clearly look like an aging profile, or the other spectrum of fitness, high fitness. So we sort of have for us a control in this sense to look at all the end of this, two ends of the spectrum. So that's the muscle disuse. So what we typically do in our lab, we take in muscle biopsies from people. This is the GeneSmart study, gene skeletal muscle adaptive response to training. Currently have more than 150 participants in this study, with several data sets and several points that we take in or collecting biopsies. We train those people several weeks, typically between 4 to 12 weeks of high intensity training, taking biopsies, and of course we are collecting all the physiological data before and after, VO2 max, peak power, and a lot of other physiological data. So the first thing that we did before we got into the exercise data, we looked at EWAS, Epigenetic Wide Association Study, meta-analysis of age in skeletal muscle. So we wanted to look and see whether those CPG sites, whether the methylome is changing because of age, purely because of age. Okay, so we did look at that, and you can see on the right-hand side or on the red, you can see that CPG sites or DNA methylation sites, the change or increase the methylations, hypermethylated, just because of age. And on the left side, you can see in blue, you can see the sites that are reducing their methylation just because of age. And it's quite widespread. More than 40,000 DMPs, differentially methylated positions or CPG sites, are changed just because of age. So that's the first step for us to know that, you know, the methylome is changing because of age. Let's, in this specific method, let's see what's happening with exercise. We also looked at transcriptome and proteome with other data sets and tried to correlate and integrate those data together. And we found 57 genes that are common between all those omic layers. This paper was published last year in JCSM. That's the first step before we're looking at exercise. We also created a website called MetaMeth or metamethylation, which you can actually look at your gene of interest and see how it changed with a gene or gene of interest or protein of interest, and how the integration between omics goes. So I encourage you to go and find this. Just press on Google MetaMeth and you will find it and see how it works. So it's really nice, freely available again, and we strongly believe in open science. So we are, you know, fostering the open science and trying to have everyone look at this website and our work as well. All right, let's come back to the exercise. And the first step, as I remind you, was the baseline. So does regular exercise or long life exercise is associated with DNA methylation aging in human skeletal muscle via the EWAS. So the first step was to identify omic data sets with physical activity or fitness phenotypes, and then to look at data sets that are following exercise training or inactivity, which is not easy to find human muscle samples in all omic layers. We looked at cross-sectional and then longitudinal interventions. We use open access repositories and our own lab, the GeneSmart, and our collaborators. So this was quite a big undertaking by Sarah Voisin, big data mining. DNA methylation and mRNA, you can see the data sets that we dipped into. And across the data sets, we have more than 500 DNA methylation data sets, samples, and almost 1,500 transcriptome, which is more, more studied in the last few years. And this is the data sets that we looked at. Yeah, so we performed meta-analysis in DNA methylation and transcriptome, and we didn't look at proteomics just because we didn't have enough data. So if anyone in the crowd or anyone that you know have proteomic data on exercise in skeletal muscle, please contact us. We're very keen and very interested with that. We have some data on that, which has been bioarchived recently, and we'll have another paper on proteomics and exercise soon. But we need the big numbers. So the next step was to test the association between age-related CPG sites, mRNA, and fitness exercise or inactivity in each individual study. Then we meta-analyzed everything to see if those CPG sites with aging are different or changing differently than the one that are changing with fitness. And I will try to explain that in a minute. This is an unpublished study, and hopefully will be published early next year. So, results. Higher aerobic fitness is associated with younger omic profile. On the left-hand side, you can see the methylome, and you can see a clear negative correlation between DNA methylation changes with age and DNA methylation patterns that are changed with exercise, and I will try to explain that. On the y-axis, you can see that VO2 max and on the X, you can see the age. These are CpG sites in the transcriptome, in the methylome, and genes in the transcriptome. And those CpG sites, these are CpG sites that are reduced with age, reduce their methylation or gene expression with age, and increase their VO2 max. Okay, so negative correlation between them. And as you can see here, we see a beautiful negative correlation in both omic layers with an FDR of 0.05 in both methylome and transcriptome. And at some CpG sites, and that's really interesting, and transcript, affected by both age and VO2 max, we see that every increase of, you can see here, every increase of 1 milliliter minute kilogram in VO2 max is can be explained by 2.9 or 1.9 years. Okay, so every increase in 1 unit of VO2 equivalent to reduce of 2 or 3 years of age. But this is, of course, only one or specific sites in the methylome, and I should state that it doesn't happen in the whole genome and only in some CpG sites. But it's remarkable and amazing that there are CpG sites and DNA methylation sites that can actually increase, with the increase of VO2 max, can shift the pattern in a way that you look almost 2 or 3 years younger. So I hope I explained it properly, because it's a bit of it's a bit of hard thing to explain, but also to understand, just because it's heavily statistical. We used 3 independent cohorts in the methylome and 4 independent cohorts in the transcriptome, 350 to 400 participants, which is quite good in muscle. When we looked at exercise training, does it mitigate the aging, the omic aging? Then we weren't very statistically robustly powered in the methylome. We did see very nice trend. Okay, you can see it here. It wasn't quite significant. In the transcriptome, because we had almost triple of the sample, we showed significant reduction or negative association between, again, exercise and CpG size that are changed with age. So they kind of negate each other. Then we looked at muscle disuse. Does it accelerate transcriptome and methylome? Now, unfortunately, we don't have, and I'm not aware of any data set with this model in the methylome, but we did have access to one data set in the transcriptome, and we show exactly the opposite. So what happened with aging is that you can see here, when we looked at the muscle disuse, we see that it actually promote aging. And physiologically, is the opposite from exercise. And the DNA methylation that we showed a positive correlation between aging, related CpG sites, and inactivity, going in the same direction of aging. So hopefully I made myself clear here. So maybe this one is a good way to show that as well, or even a better way. So if I summarize the data, you can see that each row here, just gonna try here. Okay, each row here is a CpG site or a site across the genome. And in blue, we have CpG sites that decreased the methylation with age or muscle disuse. Whereas in red, you can see that CpG sites that increase with fitness or following exercise. So again, contradicting each other. You can see here, CpG site, decrease with age, increase with VO2 max, but also increase after training. And as you can see in the transcriptome, beautifully, you can see that age and muscle disuse are going in the same direction. in those DNA methylation sites, whereas exercise or higher VO2 max at baseline and training are going in a different direction. And it's really nicely across the human methylome and transcriptome as well. All right, so these cohorts are, as I said, difficult to get. So if anyone has human cohorts with biopsies and exercise in South Africa, we'll be very happy to collaborate and very happy to hear from you. We are now performing integration to pinpoint the top genes that are related to those changes in the methylome and transcriptome, because ultimately we want to pinpoint the genes that are most predictive and most important in these processes, and then target them and try to do some functional work with using vector to methylate the promoter of genes and also sRNA. And one of our difficulties we have is that we are not aware of cell culture model that can mimic long-term exercise. We are aware of cell culture models that are, very short and that can mimic, I guess, short bout of training, but not long-term exercise. So this is the difficulties that we have with human studies. And as you can see here, the omic integration and the CPG sites, again, age and inactivity are going in the same direction, both at the methylome and the transcriptome. Yeah, so again, the functional validation of top genes, this is what we're looking at, the unmethylated promoter versus the methylated promoter to see if those genes are really predictive of aging. So that's the future of what we are doing at the moment. I want to touch on one last point that is related to other DNA methylation patterns in the human genome. So those DNA methylation patterns that are spoken about it today, called DMP, differentially methylated positions. And they're the one that everyone is looking at. But in fact, there are many, many other DNA, not many others, but some other DNA methylation patterns across the genome that we hypothesize that are more predictive of biological age. So this is Kirsten Seal, a former student of Alison. I'm sure that Alison now is smiling in the crowd over there. She's one of my best students, and I'm very thankful to Alison and South Africa for raising her and brought her to Australia to work with me. She's very good in bioinformatics, and she published this year a wonderful paper in Nature Reviews Genetics, and there are more papers to come in big journals, which I'm hoping to be early next year. So Kirsten is looking at what we call VMPs, viable methylated positions. And without getting to too much explanations, those sites in the methylome are more chaotic. So they're not changing in the same directions like DMPs during aging. They are being more, and they capture differences in DNA methylation at older ages. They are being more, I guess, chaotic is a good word, and more viable. And they change the viability with age. So Kirsten is hypothesizing that those changes are more important biologically, and she's currently undertaking a big study with more than 50,000 samples across tissues to look at those VMPs across all human tissues and to try to integrate and to pinpoint those VMPs that are, I guess, tissue specific, but also shared between tissues. Okay, so if I'll summarize my talk today, so age has profound effect on skeletal muscle epigenome. We showed that and published that. Individuals with higher fitness at baseline have younger DNA methylation and transcriptome profile. Again, we showed that across many human samples. Exercise training shift DNA methylation profile. It wasn't significant, but it does shift. We just need more samples, but it definitely shifts the transcriptome towards that of a younger profile. We also identified some of the top genes associated with the interaction between aging and exercise in muscle, which I didn't show, I didn't specify them today, but we do have them. We are now validating some of those genes using methylation vectors and siRNA just to make sure that we have causality over there. And those genes are really predictive or changing with aging and are important to exercise.